Everyone has heard of, and potentially used in their everyday life, things like vaccines, corn, and even pills. However, not many people understand their similarities or their importance. All of these things can be used for biotechnology, but we'll get into that a little bit later. There are two main categories of biotechnology: genetic engineering and cloning. There's also techniques that combine both of these categories to make a finished product. Let's start with the genetic engineering. Genetic engineering is the deliberate modification of the characteristics of an organism by manipulating its genetic material. Genetic engineers first start with the cell. In order to use the DNA inside, they have to free it. They take detergent and it pulls apart the lipids and proteins that make up the membrane surrounding the cell and nucleus. Once these A restriction enzyme, or an enzyme produced by certain bacteria having the property of cleaving DNA molecules at or near a specific sequence of bases, is taken from a bacterial cell. The enzyme cleaves the DNA at certain sequences through both nucleotide strands, leaving ends open where they were not before. These ends moving away are called sticky ends. In the meantime, the usable portion is set aside as a bacterial plasmid enters the scheme. A bacterial plasmid, or small circular strand of DNA, is taken from a bacterial cell by many ethanol precipitation. The restriction enzyme then cleaves the plasmid at corresponding bases. An enzyme called DNA ligase facilitates the joining of DNA strands together by catalyzing the formation of phosphodiester bond. It bonds the human gene and the bacterial plasmid together, forming what is called recombinant DNA. Cloning is replicating a fragment of DNA so that there's enough to analyze or use in protein production. The three main types are gene, DNA, and organismal. In gene cloning, a gene of interest is located and copied out of DNA extracted from an organism. DNA cloning uses PCR, which we will demonstrate later, in a DNA vector to create many copies of the same DNA strand. Organismal cloning, or reproductive cloning, is a method used to make a clone or an identical copy of an entire multicellular organism. Stem cells can also be used in cloning. Adult stem cells taken from a specific part of the body can only be used to develop a cell from that family. For example, if an adult stem cell is taken from muscle tissue, only a muscle cell can be made. Embryonic stem cells, however, no matter where they are taken from, can be used to create any type of cell such as muscle, nerve, or skin. The final concept of biotechnology are the techniques used to make it all possible. Gel electrophoresis is used to separate DNA fragments according to their size. DNA samples are put into wells at one end of the gel and an electric current is applied to pull them through the gel. DNA fragments are negatively charged, so they move towards the positive electrode. Smaller molecules are also synthesized from the recombinant bacteria we discussed earlier. Lastly, the PCR, or polymerase chain reaction, is used to produce multiple copies in DNA cloning. PCR takes a strand of DNA and heats it up, then takes the primers and the nucleotides to form two new strands of DNA. So what is this all good for? We all know world hunger is a problem. It may seem like a bit of a stretch, but biotechnology has the ability to change that. Scientists can use genetically modified organisms to make simple foods more resistant to parasites or diseases, allowing for more production. This would allow parts of the world with little to no food to gain access.